Hello, good students, and welcome for our lesson. We are still looking on this topic, genetics. I had already introduced genetics. We did look at the definition of genetics. It's one of the branches of biology that was mentioned to you in your when you began to learn about biology in Form 1. So we went ahead and looked at genes, DNA, the RNA, the differences that do exist between the DNA and the RNA molecule. And we also looked at the role of the DNA. One of the roles was a storing of genetic information in a coded form. The other one was the transfer of genetic information. And then the other one was translation of genetic information into characteristics that will eventually be translated into characteristics of an organism, that is through protein synthesis. So today, we want to look at another new concept, that is DNA transcription. What is DNA transcription? Maybe you can begin by defining what is DNA transcription. Then you can move forward and look at some of the events that do take place until the DNA transcription is done. Okay, DNA transcription. So this is the transfer of genetic information from the DNA in the nucleus. It's the transfer of genetic information. This is the transfer of genetic information. This is the transfer of genetic information from the DNA in the nucleus to the ribosome. From the DNA, that is the deoxyribonucleic acid, from the DNA in the nucleus to the ribosomes, which are normally present within the cytoplasm through a very important type of RNA known as the messenger RNA. To the ribosomes in the cytoplasm through the messenger RNA, through the messenger ribonucleic acid, through the messenger ribonucleic acid, abbreviated as mRNA. That is DNA transcription. So, that means therefore for a transcription to be done, which will be preceded by DNA translation until we have the characteristics of an organism, then there must be some sequential events that takes place within the DNA molecule. That includes unzipping itself for the messenger RNA to be formed. So normally the DNA molecule is the one responsible for protein synthesis in cells. And it is very important because it determines the structural characteristics of an organism. And before that happens, there are two types of proteins. One is structural proteins that determines the characteristics of an organism, and then the other one is the functional proteins, that is the hormones and enzymes, which are metabolic regulators. So in this context, we are now being specific on structural proteins that will be done by the DNA molecule through the messenger RNA. So normally, these characteristics are normally determined by a sequence of bases along the DNA strand. So there exists a sequence of bases. So a sequence of bases along the DNA strand, codes for the amino acid, codes for the amino acids to be formed during protein synthesis. Students, you do remember that the building blocks of proteins is the amino acids. So within the DNA strand, there is a way in which the bases are arranged within the DNA strand. That is, they normally form the nucleotide bases. So these bases include the adenine, guanine, cytosine, and we also have uracil for the case of the RNA. So there are normally four types of nitrogenous bases. So they normally appear in triplet base. So the triplet base, which you normally call them the codons. This is normally a set of 3 bases. A set of 3 bases, codes for the type, or codes for the amino acid. Codes for the amino acids to be formed, which will eventually be used during protein synthesis. So, having said that, that means there must be formation of the messenger RNA. So let us now see how the messenger RNA is formed and what event will proceed thereafter the messenger RNA has been formed. So formation of messenger RNA. Formation of the messenger RNA. So in formation of the messenger RNA, the following happens. So number 1, the DNA molecule unzips or unwinds itself, hence making bases free within the strand. Making the bases free within the strand. Then number 2, one of the DNA strand, acts as a template. One of the DNA strands acts as a template for formation of a new strand. For formation of a new strand, and that is now the messenger RNA. The messenger RNA. So the event that takes place until we have the messenger RNA. Maybe if you can now look at it diagrammatically. So if this is the DNA strand, for example, this is the DNA strand. Remember, it has a double helix structure, so let's assume that it has uncoiled itself or it has unzipped. So we shall have such a structure. So after it has unzipped itself, we shall have such a structure. Remember, it has 2 parallel strands. So we have such a structure. So it has uncoiled itself. And we have 2 strands. So this is the DNA molecule. That is the DNA molecule. Remember, within this DNA molecule, there is a way in which nitrogenous bases are arranged. We have several sets of nitrogenous bases arranged in that manner. We have several sets of nitrogenous bases. You want to see how? In case here we have adenine, then here we will have thymine, adenine, thymine, cytosine, we have guanine, cytosine. In that manner, adenine reacts with thymine. In that manner, cytosine, guanine. Because you have guanine here, we still have cytosine here. Because you have thymine here. Here we have adenine. We have adenine. In that kind of sequence, cytosine here we have guanine. In case here we have guanine, on this other side we have cytosine, adenine, thymine. These are some of the sets of nitrogenous bases within the DNA molecule. So once it unzips itself, then one of the DNA strand, let's say this is now the DNA strand that will act as the template. Let's assume this now acts as the template for the formation of the messenger RNA. So we have the template here. What is the template? So that means we shall be having another adjacent strand here. So remember, this is happening within the nucleus. Remember, the DNA is found within the nucleus. So in the formation of the messenger RNA strand, one of the parent DNA strand will be used as the template for the formation of the messenger RNA. And remember, RNA, for the base adenine, is replaced with uracil. So in case here we have adenine. In case here we have adenine, for this case here. So adenine, for this case, adenine will not pair with thymine because you don't have the base thymine in RNA, but it is replaced by uracil. So here we shall have uracil. Here we shall have another uracil. Of course, guanine will remain. Guanine, cytosine pairs with guanine. Then thymine pairs with adenine. Thymine pairs with adenine. Adenine. Here we have cytosine because here we have guanine. And then here we have guanine, we have cytosine, and lastly we have uracil. So you can see that we don't have any thymine nitrogenous base because in RNA the base thymine is replaced by the uracil. So now we have the messenger RNA. So this is the messenger RNA. So for the formation of this messenger RNA, one of the DNA strand was used as the template. So this is the DNA strand. That is our DNA strand. So this messenger RNA remember this is happening within the nucleus. And for this protein to be synthesized, the messenger RNA will be able to carry this information in form of base triplets, that is the codons, which will code for a sequence of specific amino acids to be assembled for the protein synthesis at the cytoplasm. So therefore, this messenger RNA will carry this information. So it will move with this information from the nucleus, and it will be able to carry it to the cytoplasm. So transfers the information. It transfers the information from nucleus to the cytoplasm. Sorry, to the ribosomes from the nucleus to the cytoplasm along the ribosomes. Along the ribosomes. Remember the role of the ribosome? It gives site for protein synthesis. So once this messenger RNA has this coded information from the nucleus, it will be able to transfer this information from the nucleus to the cytoplasm and assemble them along the ribosomes, which will eventually be used for protein synthesis. Protein synthesis. And to do protein synthesis, that is now when we have the structural properties of an individual. Well, so that is the formation of the messenger RNA. So once the messenger RNA has left the nucleus with the information transferred to the cytoplasm, assembles the coded information along the ribosomes, now we shall be having another type of RNA known as the transfer RNA. So this transfer RNA has a specific role. This transfer RNA. So once the coded information is within the cytoplasm along the ribosomes, then another type of RNA, the transfer RNA, will use the genetic coded information from the DNA in form of codons. So another another type of RNA. That is the T. So we use the small letter T, tRNA. Uses the coded information. Uses the coded information from messenger RNA. So this coded information will be used by the transfer RNA that has been assembled along the ribosomes, that is within the cytoplasm, to be able to form protein polypeptide chains. To assemble specific amino acids, to form a polypeptide protein chain. That is now the characteristics of an individual and its progenies. So that now is what is known as translation. So this is translation. This is translation. So transcription, messenger RNA carries the coded information from the DNA, from the nucleus to the cytoplasm. Then once in the cytoplasm, the coded information from the messenger RNA will be used by the tRNA, transfer RNA, to be able to assemble specific amino acids to be able to form the structural properties. That is now the polypeptide chain, and that is now translation. And that is how the transcription process is done. So this assembling of amino acids in ribosomes to form proteins using the mRNA codons is now what is known as translation. So in simpler terms, this is assembling of amino acids. Assembling of amino acids from messenger RNA in form of codons, or the triplet base. That is now known as the translation process. So eventually, it is now the protein that will be formed that will determine the inherited characteristics in an organism. That is all about the transcription process and translation. So now moving forward, they are now conversant with DNA replication, DNA transcription, DNA translation, and some of the differences between the DNA and the RNA molecule. Well, students, let us now move forward and look at some of the terms that we shall come across in this topic of genetics, which are very important before we begin by looking at the Mendel's first law, or the law of heredity. There are some terms that a student must be conversant with. So these terms are very important. So there are like 16 terms that we need to understand before we start drawing the genetic process and using the Punnett square for that matter. So terms used in genetics. Terms used in genetics. Terms used in genetics. Terms used. Let us start with the simpler terms as we proceed to look at more complex terms. So the first term is genotype. We have genotype. Genotype is very important in understanding genetics because it will be used several times. So this is now the genetic constitution of an organism. This is the genetic constitution of an organism. The genetic constitution of an organism. Of course, it is not visible. You cannot be able to tell the genotype of a person, not unless one uses the genetic process. Then number 2, we have phenotype. Phenotype. This is the outward appearance or the physical appearance. Is the outward or physical appearance. This is the outward or physical appearance of an organism. I.e., when you look at an organism, you can say whether it is tall or short. So we have tall. Examples here, we have tall, dwarf, etc., etc. Also the skin color, one can say one is brown, one is black. That is the outward appearance of an organism. Then the other term is number 3. We have allele. Allele, also known as allelomorphs. Allelomorphs. So we have the allele or the allelomorphs. So allele, this is the alternative form of a gene. Is the alternative form of a gene that determines inherited characteristics of an organism. So that is the alternative form of a gene that determines the inherited characteristics of an organism. Then the other term, number 4, the other term that shall be looking at is F1 generation and F2 generation. So we have F1 generation. F1 generation, or simply the first filial generation. Then the other one is the F2 generation. F2 generation, or the second filial generation. These are terms that we cannot avoid when it comes to genetic process. Let us start with the F1 generation. So F1 generation, this is the generation obtained after crossing or after the first cross. Is the generation of offsprings. This is the generation of offsprings obtained after a first cross. So after that first cross, the set of offsprings that will be displayed, that is the F1 generation. Then F2 generation is now the generation obtained after crossing F1 generation. This is the generation. Obtained after crossing F1. So the first cross, we get F1. Then for us to get F2, F1 generation must be crossed. So therefore, that means crossing F2 generation will give F3 generation and so on. After crossing F1 generation. NB. That means crossing F2. Crossing F2 generation gives F3 and so on. That is the filial generation, F1 generation, F2, and depending on the type of process, we can even get F3, F4, and so on. The other one that we need to understand, number 6, is diploidy. Diploidy. So diploidy, this is normally abbreviated as 2N. Here we have 2N. So what is diploidy, students? So diploidy is a condition where a cell has 2 sets of chromosomes. A condition where a cell has 2 sets of chromosomes. This is a condition where a cell has two sets of chromosomes. So normally, the diploid forms are the somatic cells. E.g., the somatic cells have diploid number of chromosomes. Somatic or the body cells. Then number 7, another term is haploidy. Haploidy, this is N. So haploidy, this is a condition where cells has only one set of chromosomes. A condition, this is a condition where a cell has one set of chromosomes. I.e., here we do have the gametes. So all gametes, or the reproductive cells, or the sex cells, have haploid number of chromosomes. Then in case we have more than the required number of chromosomes, then we shall be having what we call polyploidy. So polyploidy, this is a condition where a cell has more than the required number of set of chromosomes, that is the 2. So this is a condition where a cell has more than 2 sets of chromosomes. A condition where a cell has more than 2 sets of chromosomes. Has more than the required number, that is the 2 sets of chromosomes. So therefore, the organism in this matter can have triploid number of chromosomes, tetraploid, pentaploid, and so on and so on. So we can have triploid, e.g., we have triploid, that is 3N. You can have pentaploid, that is 5N, and so on, depending on the number of chromosomes that are available. Number 9, another term that we need to be conversant with, number 9 is what we call hybrid, and as a result of hybrid, we then have hybrid vigor. So these two terms normally confuse students. So let us start with hybrid. When it says that this is the hybrid of these cross, what do we mean? Hybrid, this is now the offspring obtained after crossing two superior organisms. This is the offspring obtained after crossing two superior organisms. Therefore, in case two organisms that are of much higher quality are crossed, then the resulting offspring will be having much improved qualities as compared to the parent. So that offspring is now the hybrid. So this performance or increased performance that is normally portrayed from this hybrid is now what is called hybrid vigor. So number 10, we have hybrid vigor. Hybrid vigor. So hybrid vigor, this is now the increased performance of the offspring that the parents increased performance. This is the increased performance from the offspring. This is now the increased performance from the offspring or of the offspring than the parents due to crossing two superior parents. Than the parents. That is normally portrayed due to crossing of two superior parents. Two superior parents. That is number 10. Then the other term that we need to understand moving forward, that is now number 11. So number 11, the other term that we need to understand number 11. So we have talked about genotype, phenotype, allele, F1 generation, F2 generation, diploidy, haploidy, polyploidy, hybrid vigor. Then we have other one, that is the back cross. Now, back cross, and some students normally confuse between back cross and test cross. You have back cross and test cross. So back cross is crossing. This is the crossing of an offspring with one of the parents to determine unknown genotypes. Of course, back cross cannot be done in human beings. That goes against the ethics of human beings, but you can have back cross in animals, you can have back cross in plants, but not in human beings. Then test cross. Test cross, this is a crossing. It's crossing of an individual with or of unknown genotypes, with a homozygous dominant genotypes to determine the unknown genotypes. To determine the unknown genotypes of a given of that offspring whose genotypes are unknown. So that marks the end of our lesson. So I believe you'll be able to look at some of these terms, analyze them, and understand them before we now look at Mendel's first law of inheritance, or first law of genetics, also known as the law of segregation. So in our next lesson, we shall be able to look at the other terms left before we now proceed to look at first law of heredity. So thank you, students, for being attentive throughout the lesson until we meet in our next lesson.